Hi everyone, my name is Sanya Sitlani. I'm an undergrad at the University of California, Berkeley. And this summer I was working in the Gore Lab with my mentor Sam Hurt on my project, Genomic Prediction of Monolignon Composition in maize hybrids. So maize is a really important crop economically and culturally in the United States. Maize stover is the biomass that's left behind after grain harvest, and it's a really important potential resource for ethanol and for producing other chemical products. But it's really difficult to break down. And one of the major bottlenecks in that is this polymer called lignin, which is a really complicated polymer. It's made up of a bunch of different components. Mainly, they're called S, G, and H. And what my project looked at was the ways that like lignin content and the composition, so the ratio of the S to G units, varies in different maize hybrids. So the goal of my project was, given the importance of lignin for plant health and economic value, we aim to test the accuracy of genomic prediction in a hybrid maize population for lignin and monolignon ratios. So genomic prediction itself is essentially a way of speeding up our selection of maize hybrids or any other crop hybrids for basically to increase the rate at which we can produce selected varieties or like improve the performance of a trait in a given variety. It uses a bunch of different models. We tested 2 of them. And for our data, we started with samples of the maize stover from 443 different hybrids, as well as genomic data that went along with those samples, and then used those to perform genomic prediction. Overall, for our results, first off, just looking at the variation, we found significant variation in both lignin and SG ratio across our population, which is really good because it means we can keep moving on. There actually is variation. We also found fairly high heritability values. They were about twice what they were for yield, which is great because that means that the phenotypic variance that we see actually can be explained by the genotype of the plants, which is good because it means we can actually do genomic prediction. For the genomic prediction, we found that number 1, both our models performed the same, with essentially no significant difference. And number 2, they both performed with moderate accuracy, which is pretty good but can be improved. So in terms of the improvements, we looked at seeing if incorporating data that we already know, so looking at genes that we already know are part of lignin synthesis, seeing if genetic markers that are close to those genes will improve our accuracy, and it didn't, which indicates that maybe the architecture is a little bit more complicated than just a few markers relating to it. We also looked at yield and lodging to see if having high or low lignin would improve or decrease yield, and it didn't, which is also good because it means that we can carry on with trying to select for lignin without decreasing yield. It also didn't have any relationship with stock lodging, although that may just be due to some of our issues with sampling. For conclusions, genomic prediction can be used with moderate accuracy to improve lignin selection process. And also that including prior knowledge doesn't seem to improve the accuracy of our models. In further research, we'd like to test with a larger population, a more diverse population, and see if we can understand more about lignin architecture.
Okay. Well, you did a great job. Will genomic prediction work well on any trait in crops, or is it limited to certain types of traits?
So the big thing about genomic prediction is that it can only be used on traits that we already know are heritable. So that's why it was really important for us to calculate the heritability. And we got pretty high values, around 0.6 for both traits compared to 0.3 for yield, which is pretty good. Like genomic prediction doesn't work as well in yield because the heritability is lower. So any traits that are more environment-based, then heritability won't work great.
Well, you did a great job. Thank you very much.